Okay, well, welcome everybody, and thank you for coming. We're just after time, so I think we will kick off, and if there are late comers, they can slip in the back. I don't know everybody, so let me introduce myself. I'm Peter Godfrey Fawcett in the Science for Action division, and we are relaunching or trying to upgrade a bit the Science for Action activity around what we're calling UNAIDS Science Now. Two main streams of activity, one of which is that there will be a relaunch of the HIV This Month digest of literature, which is just in progress at the moment, and the first issue should be coming out end of this month. And the second stream is that we want to start 2013 and ongoing by encouraging more opportunities for interaction around hot topics, science topics that are relevant to all of us who work throughout UNAIDS. So I don't know how many people we've got sitting on the other end of the webcast, but it's been advertised to all our country offices, and there is a live webcast going on to them, so that welcome any of you who are sitting at the end of your computers in the morning or the evening or the middle of the night. And today we're starting off with a discussion around HIV prevention. So I'm delighted that we've got some excellent speakers, both from within Geneva, from WHO, and also from ANRS, the French Agence Nationale pour Recherche sur SIDA and the viral hepatitis. I think prevention, of course, as we know, has been changing rapidly. It's been a very exciting time for prevention in HIV over the past few years. And that does, of course, bring interesting opportunities for thinking about how we use research technologies. And I think it will mean that we're continually having to change our ideas about preventing HIV. So today's focus is, on the one hand, on pre-exposure prophylaxis, PrEP, which will be described in much more detail in a moment, and on the other hand, on more generally issues around HIV prevention research, particularly thinking about some of the challenges for future research questions, both practical and ethical. So with that, I'd like to pass over to our panel. And the first speaker is going to be Kevin O'Reilly, who comes from the WHO HIV Prevention Department, KPP, I think you are now, aren't you? Key Populations and Prevention, I think. Something like that, prevention. I'm afraid I don't know all the acronyms. And Kevin is going to start off by talking about setting the background of some of the PrEP issues. Kevin.
Okay, thank you, Peter. And what I'm going to try to do today in a brief period of time is to present to you some of the accomplishments, i.e. what's been learned about PrEP, and some of the challenges, i.e. what needs to be learned about PrEP, as we try to move forward to consider whether and how it's an important thing to include in our prevention armamentarium. So, if I have a slide someplace. Me? I do it? There. All right. So just to summarize again, I'm going to try to review the progress and the challenges that we face. Excuse me. One of my challenges is my first cold of 2013, so excuse me if I have to.
I'll do it.
Whoop, thank you. This excellent slide from Robin Shattuck summarizes very nicely the new avenues or the new approaches to prevention that have become possible in the last few years as we begin to understand that antiretrovirals have not only treatment benefits but also preventive benefits as well. So it's now possible not only to try to control transmission of HIV infection at the time of coitus, but also prior to exposure, after exposure or even after infection has taken place. So if we look at the different methods there, that prior to exposure, we have male circumcision, which we know is very effective when used, if we can implement that in countries. And topical and oral PrEP, which have become much more appreciated and understood in how their mechanisms and how they work as well. As well, we've got oral pre-exposure prophylaxis intermittent PrEP, which we'll be hearing a little bit more about, which less is known about it now, which is why we have here today Jean-Michel to help us think about that one. Coyly dependent PrEP, and both Manjula and Tim Farley are here if there were any particular questions about how that research is progressing and how that development is coming along. And then there's also oral post-exposure prophylaxis, which is normally is primarily being used for occupational exposures, but also can be used in cases of rape. And then there's the very exciting field of after infection takes place, early treatment to try to reduce the transmissibility of HIV infection from an infected partner to an uninfected partner. And those take various different forms, from treatment at different CD4 levels up to just treatment for all who test positive. And the evidence for all of that is being carefully screened and looked at. In the last couple of years, a number of very exciting trials have produced results, and all of these are under the rubric of biomedical prevention trials. And we've seen results ranging anywhere from like 30% effectiveness with the Thai RV144 vaccine up to more than 90% effectiveness for the HPTN052, which was the trial of early treatment for individuals living with HIV infection. In the middle ground, we have a number of oral PrEP and topical PrEP studies, which are also included, ranging from about 39% overall effectiveness for the Caprissa 004, which is a 1% tenofovir topical gel that was used pre and post coitus, up to about 70%, 75% for the combination tenofovir FTC used in oral PrEP for the partners trial. So really, what is PrEP? PrEP is short for pre-exposure prophylaxis. And by that, what we mean is it's the use of antiretroviral drugs by uninfected people to try to avoid the acquisition of HIV infection during sexual exposure. Trials have typically evaluated either oral TDF, and that's where most of the work has been done, with oral TDF or oral TDF-FTC. And there are currently trials of other drugs that are in the field and starting off as well. And trials have primarily focused on daily PrEP. There is some earlier studies that have been done recently on intermittent PrEP with different types of dosing. And I think Jean-Michel may review some of that in talking about the justification for the ARNS study, IPAGE. Topical TDF, as we talked about, has also been tried as a vaginal microbicide, and the results have been encouraging. And so far, 4 trials have been completed. 1 was stopped, and 1 has had various arms cut off or discontinued. So to some degree, PrEP is benefiting from a wealth of data, and it's also cursed by a wealth of data because not everything trends in the exact same direction. So before we look at some of those results, let's just consider for a second what it is about that's so attractive about tenofovir, or the combination of tenofovir and emtricitabine. It's interesting because it has very broad antiviral activity, and it's active against viruses both found in early infection and late in infection as well. And it acts early in the life cycle of HIV, so it can block initial infection. And it's also rapidly active, and it's this attribute of the drug which makes it interesting for the concept of intermittent use. Because even if people miss doses, they can get to a sufficiently active level of dose relatively quickly, it would seem. And macaque data do support that. Also, it's safe, and all drugs have some side effects and some consequences, but in the trial so far, the tenofovir and tenofovir FTC together have shown to be relatively safe for use for people. It has a high barrier to resistance, so that early claims or early concerns about developing lots of antiretroviral resistance from PrEP use do not seem to be showing up in the trials themselves. And there's limited cross-resistance as well. And it seems to be relatively easy to use. A pill a day, or a pill as many days as you can possibly manage it, with no interactions with other drugs, no food restrictions, etc. I wish the colors were a bit brighter on this, but there are a number of trials that have been concluded. And just to give you a key to the way this graphic, which I've borrowed from AVAC, works, all the crosses that you can see on this, I think I have the crosses you can see there, and there, indicate completed trials. So the completed trials are the IPREX trial, which completed in 2010, I believe it was, and the TDF-FTC trial, which also completed. This was the CDC trial that was completed in Botswana, also on time. The other trials that have completed are the Partners PrEP trial, which completed early, and down here, the Caprissa 004 trial, which also completed early. So those trials completed, correct me if I'm wrong, Tim, that was, did it complete? No, completed on time. Okay.
Completed as scheduled.
Completed as scheduled, right. So the interesting news that came out a year and a half ago was the early completion of the Partners PrEP trial, which was stopped early for overwhelming effect. Other trials, however, such as the VOICE trial and the FemPrEP trial, have both been stopped. The FemPrEP trial was with a trial of women, of high-risk women, and it was stopped for overwhelming lack of effectiveness. The incidence of infection in the two arms was exactly equal at interim review, so the trial was stopped. The VOICE trial, which was a complicated trial, complicated three-arm trial, was comparing the 1% TDF gel to tenofovir, and not comparing them, I'm sorry, comparing the 1% TDF gel to a placebo. It was also trying the tenofovir alone to placebo and tenofovir emtricitabine combination to placebo. And the only arm that remains of that is the combination oral drug of tenofovir plus emtricitabine. The other arms were stopped for ineffectiveness as well. So the question is why these arms have stopped, and it's not entirely clear in all cases, although strong suggestions seem to be that both the FemPrEP trial and the VOICE trial have had trouble getting the type of adherence that they wanted to the suggested regimens. This is the tenofovir gel arm of the VOICE trial, which has been stopped. Other trials which are currently ongoing are open-label extension trials, as you can see here, which are follow-ons to the completed trials of IPREX and TDF2, and the Caprissa 008 trial, which is a follow-on to try to examine on a larger basis some of the interventions that were tried in Caprissa 008, FAC-01, which is a demonstration project sort of trial as well, and then down here, new methods of administration and new drugs to try for as other formulas to use for other means of administration and other drugs to use for pre-exposure prophylaxis. These, as you can see, there will be some period of time before we can find any results from those particular trials. I just want to look at this slide very quickly because it shows over here the overall results that were found from the trials. This is only looking at the oral medications now, the daily oral medications. And it shows the overall results, the modified intent to treat analysis, but it shows over here what happens when you can screen for presence of drug in the individuals who are in the trial. And the effectiveness of the drug climbs enormously when you can demonstrate that people were actually taking the pills. So the takeaway lesson we can use from this is that PrEP, like condoms, works pretty well when it's used correctly. It doesn't even have to be used completely correctly, but sort of correctly. But it's not effective if it's not being used. And The PrEP trial, the strong indication is that was the problem that led to the closure of that particular trial, is that they weren't able to get people to use the drug as they were being asked to do. And there seems some indication that we won't know yet for another year perhaps, that that was the same case with the voice trial. Just to show you quickly that the Partners PrEP trial, which was closed early for strong effect, had a overall 67% effectiveness for tenofovir alone and 75% for tenofovir plus emtricitabine. And when you look at the graphs here, you can see that the placebo arm shows rapidly rising infections over time. The FTC combination arm shows a lower rate, and the tenofovir arm here shows lower rates of infection. So it was a well-conducted trial that shows that if people do take the drugs, and they were pretty adherent, as can be demonstrated not only by self-report, which is unreliable in this field of research for the most part, but also backed up by laboratory characteristics as well, that if people do take the drug, it does seem to work. The question is, how should people take the drug? And it's a big discussion about, are we talking about putting people on a drug like PrEP for the rest of their lives? In which case, isn't that a very expensive and dangerous thing to do? And the conclusion for many people, like Nellie Mugo, who says it beautifully at the IAS meeting in Washington last summer, that PrEP is not for life. PrEP is for a season, which is a very kind of colloquial way of saying that there will be times in people's lives when PrEP will make sense, and there'll be times in people's lives when other things will make sense or make more sense. But PrEP should be an option that's available to people at times when it would make sense for them. So PrEP can be an attractive option for people who won't use other prevention methods. They don't like other prevention methods, or something about them, it presents a difficulty. It's interesting in the IPREX data, for instance, which was a multi-country study done across 3 continents, primarily with data coming in from Latin America, that there was a large, relatively significant cohort of people at the beginning of that trial, this is all MSM, large group of people at the beginning of the trial who did not use condoms. And despite all of the interventions that were provided to them into the conduct of the trial, interventions to help them get tested and counseled and recognize their risk and use condoms, etc., all of which they're ethically mandated to provide to the trial participants, these people still did not take up condom use at all. They did, however, adopt PrEP use. So there's a group of people for whom condom use is just inconceivable or impossible or undesirable or something. It's not up to us to judge why they're not using the condom use to see if we can force them to use condoms, but it is up to us, I think, to provide them with alternatives for people who've clearly demonstrated that they're not interested in condom use for some reason. There's also, for some people, even though we know that early treatment can block infection from an infected to an uninfected partner, there are hesitance on the parts of some people to accept early treatment as well. Other people might want to exercise more primary control over their prevention methods than trusting it to another individual to take early treatment, perhaps. And some people are in situations or circumstances where they're just really not, condoms and other prevention methods are just not an option for them. For instance, people in closed settings like prisons, or HIV-negative women who are in a serodiscordant relationship and seeking pregnancy, they cannot possibly use condoms. So we have to recognize the multiplicity of needs that people have, and following on the model that was developed over time for family planning, the more options we give people, the more prevention people actually do use. For instance, from the Partners PrEP trial that was done in Kenya, they asked a survey question for people in serodiscordant couples, because Partners PrEP was a serodiscordant couple study. They asked the infected partners, would you be willing to take treatment? Because that would be the first option for people. Would you be willing to take treatment? And about 42% of men and 30% of HIV-infected women said, no, I'm not ready to start taking the treatment, leaving the uninfected partner, of course, with the need to find some means to control exposure to HIV infection. And primarily the concerns were about, the concerns were about side effects or the stigma or the pill burden associated with the early treatment model. In addition, in HPTN052, which is a very important study, it was a very significant decline in the linked transmissions, where the transmissions were linked virally, so they could demonstrate that the HIV infection that was transmitted came from the infected partner. But if you looked at the unlinked transmissions over here, you find that a third of the infections overall that occurred came from outside that serodiscordant partnership. So the uninfected partners were having sex, unprotected sex, with other individuals who also were infected, and were getting infected by those individuals, not by the infected seropositive partner who was on treatment. So this underscores the need that even in serodiscordant couples where the infected partner is on early treatment, there may be need as well for pre-exposure prophylaxis for the uninfected partner to protect themselves. So clearly we have a lot that needs to be, we have a lot of options and a lot of opportunities with all these various means of antiretroviral-based prevention and biomedical prevention now. But we do have challenges as well. And we've got big programmatic challenges, we've got community challenges, and we've got political challenges, which all need to be addressed before we can really clearly assess how viable PrEP is as an option to recommend strongly to countries. For instance, we need to figure out how to deliver PrEP safely and effectively and acceptably for the end users who are involved. We need to figure out how to deliver PrEP to create adherent users, because clearly adherence is the major stumbling block for PrEP. We need to minimize risk of resistance, and the major challenge with that seems to be the ability to diagnose accurately at the onset of PrEP use people who are clearly uninfected at that particular time. And we need to take into consideration the needs of the people who will be using PrEP. One of the challenges in trying to measure the adherence to PrEP is the fact that we can't use a treatment model for measuring adherence. If we put people on treatment, we expect them to stay on treatment for their own health and their own benefit. If we put people on PrEP, they may stay on PrEP for a while their circumstances reflect what they need at that time, that PrEP is the best option for them, but they may subsequently switch to another method of HIV prevention, which might be more relevant to them as their circumstances change. So we can't just say people who are not taking PrEP, who were originally put on PrEP, are no longer adherent. We can say they're no longer taking PrEP, but they may be adherent to some other element of combination prevention. So it's difficult to measure exactly what adherence is going to mean in PrEP. We also have a lot of community challenges, because we need to know who are the users who are going to be can't get that right button. Who are the users who are going to be most likely to benefit from PrEP use, and what do they really understand about PrEP at this point? And there's a lot of misinformation and a lot of lack of information about PrEP that's out there right now. So there's a lot of community work that needs to be done so that people who are considering using PrEP or possibly considering using PrEP know what they're getting into, know what's expected, know how it works and how it doesn't work. And then there are also political challenges. The challenges around how to include PrEP in national treatment and prevention programs. What is the appropriate niche for PrEP? It's clearly not going to be something that would be used for everybody everywhere. But for whom should it be used? For the highest risk individuals? For closed populations? General population? This is all a national level discussion that really needs to be taken. And the problem, of course, with trying to think about highest risk individuals, which might make epidemiologic sense, is that they're often the politically least empowered people. And therefore, it's a very difficult political decision to take that you're going to invest lots of HIV prevention money in providing ARVs for PrEP to sex workers, or drug injectors, men who have sex with men, etc. And then there's also the very big question of who's going to pay for all of this? Who's going to pay for the PrEP? So it's definitely a challenging issue, and onto this crowded stage comes intermittent PrEP. Strongly justified need. If we can figure out how to administer intermittent PrEP, we can cut costs, we can provide, if we can provide sufficient protection on an intermittent basis. But there are very complicated research challenges, and there are also complicated ethical issues, and those I would not like to touch on. I'd rather leave that to Jean-Michel.
Kevin, thank you very much. And I think rather than take questions to Kevin now, I'd rather save that for the discussion period because we're already eating slightly into our time allocation. So I'd like now to introduce Jean-Michel Moliner. Professor Moliner he's the chief of the infectious disease service at St. Louis Hospital in Paris, northeast Paris, a big infectious disease unit with a large number of immunocompromised patients, not just HIV, but other immunocompromised as well. But his research has focused a lot on HIV prevention recently, and he runs also the Coordinated Action Group on trials for ANRS, the French research agency into HIV and viral hepatitis. So, and he very kindly has he's only just come back from another meeting where I think he was being asked to present what was happening with the trial that he runs in Washington, and he's only just come back from there and has hopped straight on the train and come down from Paris to present to us. So I think he's agreed to present in English, for which I'm very grateful. Jean-Michel, thank you.
Thank you. Well, thank you for your kind invitation to be here today, and thank you also for the opportunity to present to you, our thinking and the current status of our Hypergate trial. And also I was asked to address the issue around the ethics for this trial and why at the NRS we have decided to do this trial. So what I'm going to try to do over the next 15 minutes would be to give you an update on where we are with the trial, why we did this trial, and what are the ethics challenges that we had to face with this study. And I hope I will convince you that this Hypergate study is an important trial to carry out for the PrEP field. So as you may know, there is quite a strong controversy around PrEP, and I have listed on this slide a couple of issues that are involved in this controversy. Is there a need for PrEP in Europe? Do we have enough confidence in PrEP effectiveness? And Kevin has shown you the results of a couple of studies, and I will come back to that. Is the high level of adherence that is required for this PrEP strategy to work achievable? What's the risk of selecting an antiviral resistance? Is the safety good enough? What's the cost effectiveness? And an important issue also, is there a risk of behavior change that could offset this benefit, since this benefit is not that huge? So the first question is, do we really need PrEP in Europe? And the answer, looking at these data from France, looking at the rate of new HIV diagnosis from 2003 to 2008, show you that clearly there is a peak in incidence of new infections with HIV in the MSM community. So clearly we need new preventive tools, in particular in MSM in France. And this figure is right also for other large cities throughout Europe and North America. The next question, and I don't have time to cover all the issues, so I have selected a few of them. An important issue with PrEP is, do we have enough confidence in PrEP effectiveness? And what I have tried to do here is to summarize the data we have with oral PrEP. And I think what's important here is to look at the efficacy, which was 44% in IPREX, very high in partners PrEP. And as Kevin said, this was a study conducted in serodiscordant couple where the HIV-infected partner was not receiving treatment. Something which is kind of not probably feasible or appropriate in, let's say, in France or in Europe. TDF2 60%, and then we have FemPrep 6%, so no clearly efficacy, and the tenofovir arm of voice, no efficacy, and we're looking forward to see what Truvada does in this context. What's also important when you look at the efficacy of this prevention trial is to look not only at the efficacy, the point estimate, but to look at the lower bound of the confidence interval. And you can see that although all these trials, only in partners PrEP, you would actually cross the 30% bound. And this 30% bound is thought to be important for public health benefit of these preventive strategies. Indeed, when you look at the other prevention trials that have been done, PACTG 076 in pregnant women, or the 3 circumcision trial, First you have, let's look at the circumcision trial, very high consistency in terms of efficacy, 60% in all these trials. And if you look at the lower bound of the confidence interval, it's always at least above 30%. So this is not what we have right now with PrEP. So what are the differences between these trials? I mentioned the consistency of the results with the circumcision trial. We have a single trial with PMTCT, but the p-value was very high. I mentioned already that all these circumcision trials excluded an efficacy below 30%. Also, which I think is quite important, is that circumcision and PMTCT are one-time interventions. And for daily PrEP to be effective, you have to have efficacy of the regimen first, but also a full adherence to daily use. And if you have a regimen that has efficacy, but for which adherence is not good enough, then the effectiveness is going to be low. And for example, condoms, we know, are not used daily unless there is a daily sexual intercourse. So my thinking about that is that daily PrEP is a nice proof of concept that PrEP can work if you take your drugs daily, but I don't think this is a viable strategy for implementation. Kevin touched a little bit about this issue of adherence. If you look at the efficacy in trials according to adherence, you have different results according to what you're calling adherence. If you focus on pill count or self-reported adherence, you could see that actually adherence was quite high in all trials. If you look now at adherence measured by the proportion of patients who had plasma drug level detectable, then you could see a difference from only 37% in FemPrep up to 82% in Paris PrEP, and iPrex is somewhat in the middle with 51% of adherence according to plasma drug levels measurements. Another way to look at the iPrex data is to look at the incidence and efficacy with adherence measured by pill use. And it's only when you reach an adherence rate above 90% that you have an efficacy in iPrex that is an effectiveness that is 68% with a confidence interval that crosses 30%. So that means that in the setting of iPrex, a study performed in MSM, mostly in Latin America, Thailand, South Africa, you have to ask people to miss fewer than 3 doses a month. So how to improve adherence with a daily PrEP regimen? Well, we've seen much higher adherence in serodiscordant couple, and there are many explanations for that. The first would be that the uninfected partner sleeps, I would say, with HIV every day or every night. So there is an incentive to take daily PrEP, and also your partner can support you with adherence because he doesn't want to infect you. On the top of that, within a serodiscordant couple, you have a low incidence of STI. You are not exposed to patients with primary HIV infection with very high viral load in their semen, for example. So how to try to improve adherence in trial? Well, we know that self-reported adherence pill count may not be reliable. We may use plasma drug levels so that you have patients with daily PrEP, you measure drug level is not good enough, you will tell your patients you have to do better with adherence. But we know that even plasma drug levels, when you talk with people who do that a lot, is not fully reliable because the patients can just take a pill a few hours before showing at the visit, and that will tell you that maybe actually these patients was adherent, and in fact he was not, probably. So we can try to provide better support for adherence, but I think that we need to assess more friendly regimens for long-term use, and intermittent PrEP is probably one of them. What do physicians think about HIV prevention? This was an interactive clinical case in the New England Journal of Medicine that was presented in August, just after all PrEP trials were published in the same journal. And the readers were asked about their position recommending PrEP or do not recommending PrEP in a 46-year-old MSM with multiple sexual encounters, a recent HIV negative test asking whether he should or not receive PrEP. And the answer from more than 1,000 physicians from a number of countries was that only half of them were in favor of initiation of PrEP because they thought that it was difficult to alter sexual behavior and PrEP should be embraced as an additional tool. But those against PrEP were concerned about the risk of sexual behavior that could offset the benefit, resistance, the low effectiveness because of the low adherence. What do people think about that? And this is a survey we did in France among gay men in July 2012, so it was after FDA approval, after we got all the results from this trial, among near 1,000 of gay men. This is on the internet. And actually, the awareness was quite low. Only 30% have ever heard about PrEP. Only 12% felt they were well informed. In terms of readiness to use PrEP, only 17.5% thought they were going to use PrEP if the effectiveness would be 50%. And it's only if the efficacy would be at least as high as 90% that 50% of these gay men would consider using PrEP. Clearly, gay men would prefer on-demand PrEP rather than daily PrEP. A large majority would prefer on-demand PrEP. Those more interested by PrEP were those with unprotected anal sex. For these people, the risk of adverse events with PrEP is a major concern. And in terms of risk compensation, 42% thought they might reduce their condom use, and 27% even could stop their condom use with PrEP. So clearly, there is a lot of work to do in this area. What about intermittent PrEP regimens? And why this interest in intermittent PrEP? Clearly, animal models support this dosing schedule, and data from the monkey models at the CDC, but also in mouse models, have shown that you can protect in the models, as well as with daily PrEP, infection with the sheath, for example, in macaque, with an intermittent schedule. It's clearly a more convenient treatment strategy. You could expect a better adherence and a better coverage of the high-risk sexual intercourse, and clearly a better safety. And we know from the microbicide field that, for example, in the Caprisa 004 trial, where intermittent use of tenofovir gel was required, that actually this was associated with a protection of 39%, whereas when within the same high-risk women in sub-Saharan Africa, these women were instructed to use daily gel in the VOICE trial, there was no effectiveness. So sometimes less can be more. Also, we know that sexual activity is not permanent, even in gay men, and it is usually concentrated during weekends and pre-planned. And also, if proven effective, people can adapt their behavior to intermittent PrEP. Not to mention that this would be more cost-effective. So which design now to assess intermittent PrEP? A trial comparing daily to intermittent PrEP seems unrealistic, because you would need, in a non-inferiority trial, nearly 20,000 participants. This could also lead to behavior changes, and the results are going to be very difficult to interpret in an open-label basis. You would never know if actually, if the two arms perform equally, if equally is better than nothing. So that's why a placebo-controlled trial remains the best way to assess intermittent PrEP, and also to confirm the effectiveness of this strategy. And we have calculated that this would need approximately 2,000 participants, probably less, to achieve this goal. Also, in a double-blind trial, participants will not know if they are receiving an active drug or not, and as is had been shown in all the other PrEP studies, there would be, therefore, less risk for sexual disinhibition and pill sharing than in an open-label trial. So this is the design of the Upper Gaze study. This is a phase 3 study looking at the efficacy of on-demand PrEP in a randomized placebo-controlled trial. So it targets high-risk MSM with condomless anal sex with at least 2 different partners in the previous 6 months. And in this trial, people have access to a full prevention package of services. And in one arm, they would use Truvada before and after sex, and in the other arm, placebo. So in terms of prevention package, and this is clearly important, they have counseling, testing for STI, even if there are no symptoms, condoms, vaccination for hepatitis B and A, and also full access to post-exposure prophylaxis, which was not available in most of the other PrEP studies so far. So we clearly want to demonstrate that intermittent PrEP has an additional benefit, and it's not a way to substitute one prevention method by another. So where are we with hypergay? The trial was designed before Caprisa and Hyprex results were available. And we've been working with the community for the last 4 years to work to prepare this trial. The gay community involvement was quite high, and the largest advocacy group in France, UNAIDS, is a partner of the study, help us with recruitment. We have set up a community advisory board. And the decision to launch the trial early 2012 was made after the results of all other PrEP trials have been released. We have started the pilot phase of this study in 3 sites in France and 1 site in Canada. We'll start actually this month to ensure the feasibility of doing this type of trial in Europe and to secure funding. So the first patient was randomized last February, at the end of February. We have just recently got approval from the Ethics Committee in Canada, and we are partnering with the Canadian Trial Network to do the study in Canada, and we are looking forward to extend the study in other European countries. You can see here the rate of enrollment in the trial. In France, we are targeting 200 participants and 100 in Canada for the first pilot phase. So far, I would say, despite the controversy around the study, it's pretty good. And we've been also rewarded by the satisfaction of patients enrolled in the study. I mean, we've been seeing almost no loss to follow-up or discontinuation so far. So is hypergay ethical? So I thought I would show you a couple of ideas here. Hypergay has been approved by the Ethics Committee in France and Canada. It has been approved by ENRS, the Canadian Trial Network, and our DSMB is supporting the continuation of this trial as it is. It is supported by a number of gay advocacy groups in France and Canada, not all of them, but the majority of them. And also, I'd like to share with you that other ongoing prevention studies are done without daily PrEP as the standard of prevention. The VOICE trial, for example, is continuing, still ongoing now with a placebo arm with Truvada in high-risk women in sub-Saharan Africa. In Bangkok, the CDC is conducting an ongoing study in IV drug users, tenofovir versus placebo. The PROUD study in MSM in the UK is actually randomizing people to immediate versus deferred PrEP. The FAX-01 study in South Africa, it's a study comparing the tenofovir gel versus placebo to try to duplicate the Caprisa 004 results. And more recently, the HVTN 505 trial in the US is actually enrolling MSM, more than 2,000 of them, to assess the efficacy of an HIV vaccine or placebo. And in this trial, the design was adapted to the results of the PrEP studies and the FDA approval of PrEP. And participants can choose actually to use PrEP in the study, but PrEP is not compulsory or is not provided to as the standard of prevention. So as you know, WHO is recommending that daily oral PrEP may be considered as a possible additional intervention. I would like to stress that hypergay cannot be summarized as only a placebo-controlled study. It is a prevention trial where participants get free access to a full prevention package that is unavailable outside the trial. So it's important to realize that participants in both arms would therefore reduce their risk of HIV infection and their risk of acquisition of other STIs. Having a placebo-controlled trial is also a way to provide unbiased evidence to assess the potential additional benefit of PrEP in MSM in a context that is different from Hyprex, because we are targeting here MSM in Europe, not the young MSM that were actually enrolled in the trials in Latin America. This is also important to confirm the efficacy of PrEP in these different contexts. And it's difficult to assume that this is going to be a fully positive trial. Clearly, we want also to assess a more convenient PrEP regimen that could, we think, also foster the use of PrEP in general. And this trial might be the last efficacy trial in MSM. So in conclusion, I think there is a clearly urgent need for new preventive strategies for HIV. Daily oral PrEP is effective, but only when it is rigorously taken. And we need PrEP regimen with better effectiveness. Hypergay is the only on-demand efficacy trial right now. There is another HPTN 069 trial in the US enrolling patients not only in the US but also in developing countries, but it's only a phase 2 study looking at behavior, not efficacy. The success of this trial may allow PrEP, intermittent PrEP in particular, to become a public health strategy. However, we're facing new challenges. I mean, trial extension to the other European side is critical for the success of the trial, and funding is currently probably one of the most critical challenges we are facing today. With that, I'll stop here and thank you for your attention.
Jean-Michel, thank you very much for a comprehensive overview, both of the rationale and of some of the challenges that you faced in designing the study and taking it forward. And I think that's a very interesting and useful presentation. So what we've done is we've asked a couple of comments on the sort of situation so far from within UNAIDS. First of all from Jason and then from Kate. And then Stefano Vella has kindly come from the Instituto Sante Superiore di Sante. I can't say the word right. ISS anyway. It's Italian. And Stefano will lead a discussion with you around some of the issues raised. So prepare your questions and Stefano will then gather those and lead the discussion. But first, we'll ask Jason to reflect from the ethicist side, and I think we'll start off from the document that WHO and UNAIDS produced and updated last year, ethical considerations in biomedical HIV prevention trials, that lays out a number of points. So Jason, over to you.
Okay, thanks so much. First of all, I want to bring regrets from Susan Timberlake. Unfortunately, she's home sick today, but I was very glad to have the opportunity to step in also as somebody who was part of the process in 2007 of revising our ethical considerations guidance on biomedical HIV prevention. I think this is a wonderful opportunity for colleagues and also to hear from really people on the front lines of doing cutting-edge research that's really looking at how to make the best possible science more available in ways that people can practically bring this into their lives. Just going to, I was invited to say a few things coming out of our UNAIDS experience in updating the ethical considerations document. I want to just touch on a few of the key guidance points that were certainly, I think, where we saw some of the live issues, the current developments that we were seeking to address in the update. And I think we've already heard about the central role of community participation. We've seen that in medical ethics and research ethics more generally, but I think we've also seen that quite practically to run successful research, really having the ownership, the involvement of affected communities. It's important for the smooth rollout of the research. It's important for your outreach and recruitment efforts. And of course, it's also the right of people to participate in the matters that affect their life. Also trying to shape the direction, hopefully, so the benefits of science are coming back in. So the revised guidance also, in guidance point 2, made a strong point again about the central role of community participation in the design and the delivery of the research program. A major point of discussion was around standard of prevention. And basically our guidance says that the best available prevention should be made available to study participants, and this is the obligation of researchers to make this available. Now, of course, this is a standard that is generally applicable in research ethics, but of course it becomes very important when you talk about prevention research of any kind, and in HIV prevention research specifically, because obviously the people that you're looking to enroll are going to be people who are at risk and being exposed to HIV. So as you talk about what is your obligation as researchers towards those people, there was a strong consensus that making available the best science to these people, and I think as Kevin quite eloquently outlined, we know that even the best science is, we know what works, but it's not necessarily practical, viable, desired by people themselves. So again, even when making the best information, the best commodities available, we still know that people are looking for wider options. And the question is, how do we really have the recruitment that brings those people into this research? Another guidance point came back to control groups. This is, of course, bigger than HIV prevention research, but the guidance came out saying that yes, it can be acceptable to use control groups when you have uncertainty about how this intervention is going to roll out in a specific study population, i.e. you don't have data that will give you some degree of confidence about will this produce something that is beneficial with this specific population. And that was the general view on how to approach the question of control groups. Of course, it's also something that an institutional review board or an ethics, research ethics committee has to grapple with when they look at the actual design of the study and the available information. And the last point I quickly wanted to touch upon was availability of outcomes. The idea is that research should really bring new knowledge that then is going to bring that new knowledge and the commodity and new possibility back to the affected community. And I want to maybe conclude on that point as something hopefully useful to add to the conversation. I think one thing we're hearing about the general PrEP discussion is, and I think Kevin also touched upon it in the political considerations, how do we really look at this as something that is a programmatic component, something that, how viable is it to bring it in? What is the role of communities demanding this and saying this is something that we think is part of adding another prevention option here? But we know that there's incredible resistance. Of course, we're talking about key populations, and we know the resistance that existing prevention and treatment efforts have when it comes to getting the political commitment and the attention and the effort towards expanding programs there. But again, this, I think, will be, as we have new knowledge about how this is working in practice and how to make it work better in practice, how do we then take it forward to translate into things that are actually part of national programs, funded, implemented, evaluated, of course, with the active involvement of communities. So I'll leave my comments there. Thanks.
Thanks, Jason. And let's have a perspective from Kate, from the community's perspective, or from within UNAIDS, from the community's perspective, and then we'll open it up through Stefano for discussion in general.
Thanks for inviting me to participate in this panel, and apologies from Carlos Passarelli, who was supposed to be jointly presenting with me today, and I have to admit I was quite strongly relying on him for this session. I'll state up front that this is not an area in which I'm an expert, and in terms of bringing the community perspective, I got off a plane yesterday from Myanmar, having not had any internet access for several weeks, and it was only really last night at 11:30 that I sent out a sort of emergency message to a few community colleagues saying, help, what am I going to say in this session? Interestingly enough, this morning at 6 a.m. I had quite a few responses in my inbox. And 2 requests to have calls, one with New York and one with London. So it was clear that there's a lot of interest in this area, I think. And in terms of the EPO-Gay trial, it was interesting to hear some of the responses that I got. I don't think they were any particularly I didn't reach out to any French colleagues, I have to say. I didn't have time. But overall, my sense was that there was a general feeling that this trial was important, that people would like to have seen 3 arms to it, which one would be placebo, one would be one would be daily PrEP, and one would be intermittent PrEP. That would be the way that they like to see it. There was also, I think I had some good conversations with people around this idea, as you were saying, of taking daily PrEP forever, and it's not something that you'd want to do for life. And I think we've had some interesting discussions recently that certainly some of the colleagues here have been a part of in WHO and UNAIDS around B+. And putting women living with HIV who are pregnant, irrespective of CD4 count, on treatment for life, basically. And a lot of pushback, including a lot of pushback from young women who are born with HIV because of those considerations over long-term usage. And I think there's similar considerations in terms of daily PrEP. And there's my sense from the people I've talked to just today is that really it could be an important game changer. It's the word that people was using. If we found that intermittent PrEP really did have a very valuable role to play. So there is still a lot to be learned, I think, from the research that came across very, very clearly. It was also, I think some of the feedback that I got was that we also need a lot more money being put into other forms of prevention. It's not just about PrEP, and that there is the general concern over a lack of awareness amongst young gay men around not just PrEP, which was clear from yours, but other forms of prevention, and not enough education out there around prevention, not enough investment in that. Jason's touched on the whole issue of community engagement, and I think we learnt from the Thai drug user trials and whatever, and since then we've come a long way in terms of engaging communities most affected in trial design. I don't know who was involved from the community in the EPIGAY one, and my sense is it was quite broad. But with some of the friends I was talking to early this morning, it was clear that there's a lot of community who've been around for a very long time and are very used to interacting with WHO and with scientists and others. But what about the young gay man on the street, or the young community person on the street who's not maybe got that level of understanding of how trials work and whatever? And are we really hearing from them about what would make sense for them in their lives, what would work for them? Rather than just listening to the old veterans, it's really important that we're really engaging with the people who would be the end users, I think. I'm just trying to see what else came out of that. So yeah, I think key issues that people are concerned about are what happens with drug levels, what happens with resistance, what about adherence? Many of the things that you've talked about already, I think. And also, just lastly, and I'll stop in a minute, was that there was also a concern that just because PrEP has been approved in the US, doesn't mean necessarily that we should just be taking that as the way forward. And the person who said this felt that they would probably, not want to say this in public. But they were also saying, for instance, DHSS guidelines recommending use of ART irrespective of CD4 count, but, we're also, very much waiting on the results of the START trial. So, there should be some flexibility, and just because one country has approved something doesn't mean that necessarily that's completely the way to go. So, it's a few random reflections, I think, from people gathered very hurriedly. But again, just to really focus back to the issue of the importance of community engagement and really end-user engagement, and not just looking at the usual suspects in terms of how you do that design of the trials. Thanks.
Thanks, Kate, very much. I think we've taken up slightly longer than we anticipated with the presentation, so I'm going to pass over in just a sec to Stefano to encourage your contributions. I do want to reiterate that really where we're hoping that we can get input and stimulate the discussion is around two areas, one of which is what is the role of PrEP and how do we see PrEP? And you've got experts here, so here's an opportunity to ask questions and see that. But perhaps a more important question is the issue around the ethics of biomedical HIV prevention studies, which WHO and UNAIDS issued a statement on only last year. And I think a fundamental question that I want us to be pushing towards is whether we're still happy with that statement or whether we need to be working to update that. So let me just read to you the statement as it currently says from guidance point 13. Researchers, research staff and trial sponsors should ensure, as an integral component of the research protocol, that appropriate counselling and access to all state-of-the-art HIV risk reduction methods are provided to participants throughout the duration of the biomedical HIV prevention trial. So I think that this was sort of, in a sense, what triggered, that statement is partly what triggered some of the talk that was going on, saying, yeah, but then how can we be having a placebo in a trial, given that most of us accept that the efficacy of PrEP is no longer in question. The effectiveness, certainly, the ways to use it, certainly, important questions, but we need to separate in our mind important questions to ask and the ethical principles that guide the ways in which research is conducted. After all, the question was important as to what happened when syphilis wasn't treated properly, but it wasn't appropriate to investigate that question by simply following people up. So there's a clear distinction between the research question and what's important and the medical ethical framework around how that question is answered. And if we're comfortable, then these guidelines, well, we have to decide whether we want these to come back for revision, because I think at the moment there's a slight tension between the word of the guidelines and to some extent what we hear about many studies going on, and this will have implications for other studies. You heard Jean-Michel point out that one would need 10 times as big a study to be able to address the question, if you like, in a way that compared head-to-head these two different strategies, and there would still be reservations about that. Similarly, when we come to look at HIV vaccine studies, it'll mean that we can test many fewer vaccines if we say that the ethical constraints mean that everybody has to be given, unlikely, or everyone has to be offered pre-exposure prophylaxis as part of the protocol. So that's just to sort of focus with the directions that we would like to have discussion around. And I'll pass over to Stefano to make some comments and then open it up to anyone who wants to talk.
Yes, thank you very much. First of all, thank you for inviting me. Very interesting. And as you said, I hope that we'll have other possibilities to have this kind of discussions, internal discussions. Second thing is that I am actually contracted by the UNAIDS to do the HTM this month, for the moment. And at the same time, I'm vice chair of the INRS. So I couldn't be more balanced, being, in a sense, split between the two. And so before going to the questions, hopefully, I have two things just that comes to my mind when you were talking about ethics of research. When my ethicist at the school, my teacher, told me that of course, there are different ways of doing research, but the worst research, the less ethical research, is the research which is planned not to give solid data. So any research can be very ethical in terms of design, but if it's in its own will not give results that are useful for the community, that's unethical. In any case, even if the all end, something that also comes to my mind, being very old and being there since the era of AZT, we definitely need, from our lessons from the past, data that is solid. Too many times we have been trusting things that were either approved very quickly, and the data were inconsistent. And so let's say that we have some lessons from the past that we need to keep with us, saying good research is a research that gives results that are useful in any case. So maybe, do you have questions? I think it's now time for us to stop talking. And
thank you very much for very interesting discussion so far, presentations. I get one specific question. What is the effectiveness of actually taking PrEP intermittently? I mean, how many doses do you need to get in order to get good serological levels in order to have a protective effect? Do we know that? And the other issue is really this issue about, you know, yes, it would be nice to have at least intellectually, to be able to sort of have a 3-arm comparator study. However, the practical point of actually putting people on long-term PrEP, you know, daily PrEP, are they? I mean, I can think of 1,000, well, not 1,000, quite a few considerations already why, you know, this may actually in practice not work. I mean, we've talked about the cost, the availability of drugs, developing resistance, and essentially you're starting to treat somebody who's not infected with HIV, and they, in effect, until they're in the, provided they're in that, let's say, discordant relationship or whatever, they're continuously, you are becoming an uninfected people living with HIV, or person living with HIV. Anyway.
Good, thank you very much for these very neat and short questions. Do you want to answer with the same neat?
Okay, I think that's an important issue that you're raising, and we don't have the answer in humans, and that's why we're conducting this trial. The only data we have are in monkeys. And in monkeys, investigators have looked at plasma drug levels, at drug levels in tissues, active drugs, measuring, for example, the triphosphate levels of these drugs. So we know, according to what has been done mainly at the CDC, what are the best schedules that should be used in human, and that's why we have picked up, following discussion with them, actually, this regimen in hypergay, which is actually a coitally dependent regimen. And it's not only PrEP, because we are asking and stressing this point to the participants that they should also take drugs after sexual exposure. So it's a mix between pre and post exposure. So we want to cover enough time with high drug levels in these individuals with the drugs we're using for PrEP. And we also know from humans, for example, how long do you need to achieve high drug level in plasma and in rectal tissue. We have these data, not with intermittent PrEP, but we know that taking these drugs will achieve high levels in blood, but even much more higher level in the rectal tissue, for example. That's why this is a regimen appropriate for MSM.
Within how many hours?
Well, it takes less than 2 hours to achieve the peak in the plasma, and in the rectal tissue in monkeys, we have these data. It takes also the same time frame. I mean, around 2 hours. In hypergay, actually, we're asking individuals to take drugs, actually 2 pills, a loading dose of Truvada, just to ensure that they have enough level in their rectal tissue and plasma, but mostly rectal tissue at the time HIV tries to enter the body.
Thank you, because I think this is a very important element that you raised. Which is the time before I go out for this Saturday night fever. I should think that maybe tonight I will have some fun. So it's important to say that how quick it is, and it's very important. I have a question, because let me say also that the continuous, you know, if you take a continuous drug for that Sunday or for that Saturday, the rest of the week, the drug is there for nothing. I mean, it's definitely there for nothing. So that's the important point. Last issue that I have, do you offer post-exposure prophylaxis to the placebo recipients?
Well, not only to the placebo recipients, but to both, in both arms, we're providing post-exposure prophylaxis. So what we are telling these people is that we are telling them how to use post-exposure prophylaxis, when to use it, and during the time they're using post-exposure prophylaxis, they are not using their drugs or placebo. So anyway, the study is blinded, so we don't know who's taking a drug, who's taking a placebo. So, and then they resume the use of drugs.
But the post-exposure prophylaxis is not placeboid.
No,
okay,
it's not,
so now that's important. So both groups get a drug after the intercourse. So that's important. It's part of the package that you offer.
Right, and as I said, this is different from the other PrEP trials where the access to post-exposure prophylaxis was not provided. Here, and in particular in France and Canada, people have free access to post-exposure prophylaxis everywhere. In all emergency rooms, they can have access to post-exposure prophylaxis. So that's the difference. And post-exposure prophylaxis is already available in France and Canada, and we didn't want to use another strategy to replace a strategy that is currently in place. So clearly we want to know the additional benefit of this intermittent PrEP regimen in addition to what we already have at hand.
So Jean-Michel, how many of the 118 randomized so far have actually taken post-exposure prophylaxis?
Well, then this is the data from the study, and this is not like publicly available at this point. What I can tell you is that a low proportion so far has taken PrEP.
But that's my impression, is that many people don't actually take, even though they may feel themselves to be at risk, actually getting around to taking a course of post-exposure prophylaxis is quite unusual.
Anyway, this is an important issue, and we're going to look at that in details. And in particular, we want to know if being part of the study would have encouraged people to use more post-exposure prophylaxis as compared to what they did before entering the study. That would be also one of the secondary objectives of the study, to see if within this prevention package that we are offering to these participants, they would be encouraged to use more post-exposure prophylaxis.
And can I also just make one following comment to Eddie's, and then we'll come to the other question, but the comment about drug levels and the amount, there seems to me to be a slightly surprising finding from the way you presented it, that you presented the daily PrEP studies and said how extremely important it was not to miss more than 3 doses in a month. Now, is that implying that it's only on those days that you miss that you are sexually active? It's a bit surprising, isn't it, that then we're saying that actually if you miss all the days on the month, except for that time when you're sexually active, that we might expect that to be beneficial. I was a little bit surprised by that.
Right. I mean, I didn't have time to, again, go into more details. But what happened in IPREX, for example, is not that people were not taking drugs at the time of sexual exposure. They were not taking drug, period, for some of them. But
in order to get effect, they were taking drugs 90% of the time. I mean, you implied that you needed to take it. It was your comment that 3 days out of a month was.
Well, actually, when you looked at drug level in people, in healthy individuals taking Truvada, for example, you reach pretty high levels. And they have made, and published a recent paper in Science and Translational Medicine about another study of looking at intermittent PrEP regimen in healthy individuals. And they were able to show that if you take like 2 or 4 pills of Truvada a week, you would achieve the same level of drug in the plasma that was achieved in IPREX, more or less. So they infer from these data that maybe you could use only 2 to 4 pills a week instead of 7 pills a week. With our design in hypergay, for one sexual intercourse, you need 4 pills, 2 before, 2 after. So actually, we are in the same range of, providing these people enough, probably, blood levels, but at the right time, at the time of sexual intercourse. And another caveat from this analysis of, adherence, drug levels, and efficacy, when you focus in the active arms on patients with the highest adherence level, you have to be careful when you do the statistical analysis, because you should do the same analysis in the placebo arm also. Because it might be that these patients who are fully adherent are protected not because they are fully adherent, but because their behavior is different in general. And so it's a false interpretation, or not fully right interpretation, to think that those highly adherent are the best protected because they have high drug levels. That probably is one of the explanations, but it's not the full explanation. And we have examples in other studies where actually we made the wrong conclusion about that.
Jantini Jacobi, Chief Gender Equality and Diversity Division. My question mainly goes to Kevin, but I have two remarks first. First of all, I think it's very appropriate to talk about a seasonal approach. And I think that's the reality of life. What most of the time is not recognized, we are talking about irresponsible behaviors when we talk about sexual rights, but reality is different. And so I think that's a very appropriate way to position it. Secondly, for women, I think the PrEP would be a highly welcomed addition in the prevention arrangements. There's very little option at the moment, and much more research needs to be done to increase the options. Regarding the intermittent and the continuous use of microbicides, vaginal microbicides, it's actually not a surprise that there's less adherence maybe and less effectiveness of the continuous use, thinking about leakage and other problems. So my question is actually, are you planning, have you started already looking at female users' perspectives? And yeah, is there a possibility to advance on this, engaging with women's movement to make sure that there comes a demand issue also?
The issues around female users' perspectives for the microbicidal use, for the microbicidal gel, or something that Tim Farley, who's here, he's looking up, yeah. Tim Farley, who's here, can address those and what they're doing with the microbicidal use. The use of oral PrEP among women or among any group whatsoever is something that we're exploring in terms of demonstration projects, primarily in Africa, but also we're going to be looking in India at sex workers. And the first step always is to assess the awareness, the level of knowledge and comfort, and the anticipated problems or challenges that individuals who might be using the drug would expect to encounter, to see if that can be incorporated into a delivery system to try to minimize those pitfalls. So that's always the first point of departure in everything that we're trying to do, to make sure the user's perspectives are addressed.
Thank you for a great presentation, and and I enjoyed the comments. I wanted to actually take the seasonal thing. I wanted to address that briefly. I mean, we all love metaphors, but we have to be careful about using seasonal. It sounds really nice, but actually, when you're talking about a place like Kenya, and I think I heard her right, she said the season was 18 to 24-year-olds. You're talking about giving PrEP to hundreds of thousands, if not 1 million people, or some large number like that. So we have to really be a little bit more specific when we're talking about seasons. The second point or second question I had is about the other prevention part of the other prevention package, and that's actually really quite critical. And questions come to my mind, like, are you providing serodiscordant couples? Because a lot of these guys are probably in couples, or maybe they're not, I don't really know, but are you providing those people with treatment? And then how do you reconcile, when you start treatment for somebody who actually has HIV? For instance, in the United States, it's immediate. In Britain, it's less than 350. These trials would actually do quite well in places where people weren't started earlier, but what if France goes to, I think they're at 500 maybe, or what if they go to immediate? What happens to your trial?
Now, and I think Nathan was also asking, correct? And then here in the front line.
Yeah, mine was a policy point, really, and it was about this question of whether it's ethical to have a placebo arm or not. Depends a little bit on how you define standard of care in the case of the Helsinki Declaration, or state of the art in the case of the UNAIDS document. I was in Thailand in the early 2000s when the PrEP trials were derailed by civil society for the very good reason that that trial did not offer clean needles to injecting drug users, and that was very clearly, in their view, defined as standard of care for prevention. But in this case, it very much hinges on how you define what is standard of care. Is it a measure of effect? Is it that it's written in policy? Is it that WHO has guidance on it?
Yeah, I mean, basically, I was about to come on the same point that was raised just now. I mean, it's an important point that Jason referred to, but also that Peter had referred to. But then linked to that point, the question about whether the current UNAIDS WHO guidelines should be revised to on the basis of the current debate around the placebo arm of the Hypergate trial. For me, really, the interpretation is one that hinged on the interpretation of the state of care, the all available. How is it defined to be precise? The all state-of-the-art HIV risk reduction methods are provided, and then there is a second prong of the UNAIDS WHO guidance, which is that new HIV reduction methods, which will, I think, PrEP will qualify, new HIV reduction methods should be added based on consultation among all research stakeholders, including the community, as they are scientifically validated or as they are approved by relevant authorities. So I think this is also another aspect that should be taken on board as you interpret whether the placebo arm should be considered ethical on the basis of all available state-of-the-art prevention methods. So I think clearly the question is not whether we overrule the existing WHO UNAIDS guidance, but whether we have done an appropriate interpretation to use its full meaning to apply it in the context of this research that is being undertaken. And very much, thank you very much for this really interesting presentation and comment by the panelists. I think it's really good.
I think we're going to be out of time, so let's Jean-Michel.
So very briefly, actually, in our trial, stable couples are not allowed to enter the study because in France we have a strong recommendation in a discordant couple to treat the infected individual. So this is a strong recommendation to do that. So within a stable couple, with the HIV-infected one being on treatment and with the viral load undetectable, it's not appropriate to assess within a study, I mean, the additional benefit of using PrEP. So these couples are, I mean, these patients are excluded. And actually, we have excluded a few individuals because their partner was HIV-infected on treatment and within a stable couple. No, well, you know, you do what you want, but, you know, if you're in a couple and your partner is HIV infected, the best thing you can do is to have your partner be treated, and treatment is available, you know, today. Well, that's why people who have partners outside their couple can be enrolled, and that's why in the inclusion criteria we have sexual intercourse with at least 2 different partners, to avoid the risk of being in a stable couple.
Let me add, because I reviewed one thing for the audience, is that PrEP is available in France. Not very easily, I think, but it's available. So it's not a precondition to be part of the trial to access PrEP. So that's I think it's important, because you remember the old times that the only way to get an antiretroviral drug was to get into a trial and then split the drugs within your friends. So this is not the case. I understand correctly. You have PrEP available in France?
Well, let's say that PrEP is not approved. Truvada is not approved for PrEP, but Truvada is approved for the treatment of HIV infection and also for post-exposure prophylaxis of people HIV negative who have been exposed to HIV. So actually a physician could write a prescription with Truvada and the prescription will be reimbursed at this point. So it is possible. It's probably not easy, or there is not an open access to Truvada for PrEP, but Truvada is available at any pharmacy. The second question was on the
package, the prevention package. What's the state-of-the-art prevention package?
Well, so I think I already addressed that, but in our study, I think we are providing the best prevention package we can have. I mean, we have counselors from the community, actually, that were trained by one of the major advocacy groups, so people spend a lot of time for each visit. We have focus group where participants can discuss. Actually, we have had also meetings with the participants. Participants ask to have interaction with me and the other scientists in the trial. We provide free condoms, we provide testing for STIs, and this was actually quite popular because participants were happy to show that they were, clean, or they didn't have any STIs. And we were actually amazed to see the high rate of STIs we had in this trial, and most of them actually were asymptomatic STIs. And so we have been treated a lot of STIs, asymptomatic STIs, because we're using PCR to diagnose chlamydia, gonorrhea, we check for syphilis, hepatitis, etc. So we vaccinate people, and the uptake of vaccination is quite high for hepatitis A and B. What else? Post-exposure prophylaxis, I already talked about that. Do I forget anything? Obviously, regular testing for HIV, and but ART is for patients infected. We are dealing with non-infected participants here, right? The environment is actually the French guidelines are to treat everyone below 500. There is a strong recommendation to do that, and above 500 is, I would say, less strongly recommended unless you have a very high viral load, you are in a stable couple, you have renal disease, co-infection with hepatitis B and C. So actually, for not being treated is quite hard today, especially with the new regimens, with a single pill regimen today. The last question was on placebo. So I think I tried to explain why we feel a placebo arm is not only ethical, but is also needed in the study. Because right now, daily PrEP is, I think, kind of stuck with, let's look at what's happening in the US, for example. The FDA approved Truvada in May. So 7 months later, the uptake of Truvada for PrEP in the US is very, very low. And my colleagues from Gilead were actually desperate because of that. They have put huge effort to promote the use of Truvada for PrEP in the US. They showed us, on their website, you have everything you need. They provide free condoms. They provide a lot of papers so that, the prescription and the use of PrEP would be facilitated. And despite that, the uptake is so low that they couldn't even give numbers of how many people actually are using daily PrEP in the US. And on the top of that, they are facing now a lot of criticism from the community in the US about the use of PrEP. Because I think contrary to what we did in France, working with the community for the last 4 years on this issue, they are only starting to do that now, because in most of the PrEP studies, they were not done in the US, only in a few cities. So there was no wide awareness in the country about PrEP, and the discussion are just starting now. And we know that it's going to take time before people really, I'm not talking about people in the gay advocacy group that are well aware of the science, but I'm talking about people, like you said, who are going to attending the venues or going to bars and discotheques. And these people have no exposure and no awareness yet about PrEP. So there's a lot of work to do to make them aware of, what are the issues around PrEP.
Thank you, Jean-Michel. I would like to leave the floor again to Peter for Dr. Bartos to conclude.
So I'd like to make one final tiny interjection, which is another major ethical principle, is of course that of autonomy. And I think it's very clear in IPAGA that consent is there. It's not a situation of a huge imbalance of power. I think that the issues of allowing people free access to decide whether they want to be in a trial is embedded within ethical principles. Of course, in some other situations in the world, that may be much less easy to ensure. So I don't think, we certainly haven't invited Jean-Michel here to sort of, as it were, say, no, we don't think this is ethical, but I think that we need to be considering the ethics of HIV prevention. Does any of the panel have any burning things? We're already over time, so unless you've got something burning. Kate?
Just very briefly, and this is a bit of a generalization, but I'll say it anyway. I think there is an overall, certainly amongst the community folks that I know, sense that PrEP is something that we're really very much talking about in so-called rich countries, and that there isn't really a sense that it's particularly applicable elsewhere, certainly at this stage, and that it's more important to put resources into actually getting pills into the mouths of people who are already infected with HIV, and that should be our priority, as well as ensuring more money is going to other forms of proven prevention. So I'm not sure how much that would change. I think, Kevin, you were talking about specific populations. I think there's a lot of issues related to power dynamics with other specific populations, whether it's, it's women, whether it's sex workers, people who use drugs, that will be very different from, gay men in the north, I think. So I just wanted to make that point before we end. Yeah.
So is that...
Just one word to say that the work and research in prevention of HIV will benefit to the area of prevention of STI in general, not only HIV.
So thank you all very much. And I'm going to pass to Michael, Michael Bartos, our chief and leader of our team, to say at the end. Before I pass to him, I just want to thank particularly Celeste for really organising this meeting, Celeste Sandoval, and the support that she's had from Sandra, Aubrey, who's not here today, and from Angelo on the camera. We do want to have sessions like this on a rolling basis. We want topics that you think are worth discussing, so please email myself or Celeste if you think this has been useful and if you'd like to have other sessions, perhaps on about a monthly basis of this sort of nature. And I'll pass to Michael to say goodbye.
Thanks. I just wondered when Professor Molyneux told us about Gilead's desperation for the lack of uptake of Truvada, whether we actually need to issue an advisory to stock markets that we've made such comments about things which may affect the Gilead share price. That aside, let me just say, on two things. Firstly, on the ethical question, we know that if it were a simple question of saying, here is a pill, if you take this pill, it will reduce your chances of becoming infected. We're going to ask you to be in a trial where you either take this pill or you may be randomized to a placebo. We know that that would be unethical. It's clear that that would be unethical. Now, the thing is, what we have with PrEP is not something where we go to people and we say, you take this pill, it will reduce your HIV infection, because what we have as the weight of scientific evidence is continuous PrEP over a long period. So actually the question then in terms of what's ethical in terms of the placebo is not to compare just this question of a patient or a person, a participant in a trial, thinking in this one moment, I may take this pill and it will reduce my risk, or it might be a placebo. That would be unethical. What we're instead talking about is the comparison between, on the one hand, something which is either a strategy to do with continuous PrEP or, in fact, intermittent PrEP, which we don't know about. And so, of course, it's because it's those things that we don't know about that there are these questions which then become difficult policy questions as well as difficult ethical questions. And the second thing, I think, is if we look at the populations that are the populations who would be of most interest in relation to whether it's daily PrEP or whether it's intermittent PrEP, basically people who come along and say, I think I can't do anything about the fact that I'm continuously exposing myself to HIV. This is my situation. Now, the interesting thing is, of course, that as a group, as a set of people who identify themselves as putting themselves continually at risk of exposure to HIV and feeling they can't do anything about it, that's, I guess, that's the group in terms of prevention terms that we most want to get to, that we most want to say, you are people who should have intensive support for a strategy that works for you. And that's one of the reasons that, for me, I kind of think the offer of PrEP may actually be much more important than the uptake of PrEP, because being able to say to people, come forward if you think you're continually exposing yourself to HIV and can't do anything about it. And that is something that deserves to get you attention, deserves to get you support. That's, I think, a sort of an interesting notion about what the practical impacts might be. As Peter said, the intention is from the Science for Action team that we do these debates on a regular sorts of basis, that we create a forum for interaction. I want to repeat his thanks to Angelo, Sandra and Aubrey for their support in relation to this, to say that both Peter and Celeste are generating this platform of UNAIDS Science Now, which is meant to be a platform which will be debates like this, webcasts like this. I don't know how many people there have been out in our offices who've been able to watch this. 22. Well, that's a good set of countries, especially for this time of year. But we'll also be talking about interactive blog platforms as well as other ways of making sure that these debates and consideration, basically our attitude, our orientation is there is no space left in the world now for thinking we should have these difficult debates behind closed doors. We're saying these difficult debates have to be out in the open with the maximum means of participation. And that's certainly our commitment, I think, from the Science for Action team and the platform that Celeste and Peter are creating in terms of UNAIDS Science Now. Then I just want to add thanks to Jason and Kate from within the UNAIDS Secretariat, to Kevin from within the UNAIDS family for his contributions and the fact that he's been tracking and been on top of leading our response as a UNAIDS family on the PrEP issues. But then also to in particular, one of the advantages of Geneva is that we're not far from Rome or Paris, and so I do want to thank Stefano for coming here and joining us, and then in particular to Professor Molina to come and join us here. We will probably have some time for a bit of informal interaction now as people are leaving, but we have otherwise run out of our time. So thank you to all of you for your participation. Thank you.